because it tests mitochondrial substrate flux that can be affected with different pathologies or treatments. For example, today we will use it to reveal cancer cell response to metformin treatment.
It requires a minimal number of cells while having sufficient replicates and the proper control for each tested material.
The analyzer is for research use only. This technique can identify errors in mitochondrial metabolism and can be used to evaluate various drugs affecting mitochondria.
Demonstrating the procedure will be Karolína Vaněčková, an undergraduate student and research technician from my laboratory.
To begin, seed A549 cells at a density of 20,000 cells per well in columns 2 to 11 of a Seahorse XF96 cell culture microplate, leaving columns 1 and 12 empty as background wells. Fill the blank wells with an equal volume of the cell culture medium. Then incubate the cells at 37 degrees Celsius in a humidified incubator with 5% carbon dioxide for cell attachment. After 3 to 4 hours, add 100 microliters of cell culture medium in the wells. Treat the experimental group columns 7 to 11 with 1 millimolar metformin, and the control group with an equal volume of sterile distilled water. Then return the plate to the incubator. For hydrating the sensors, pipette 200 microliters of sterile water per well into the utility plate. Then carefully return.